It's my pleasure to introduce Dr. Surendra Sharma, who is originally from here in India, but we're very fortunate to have him working with us in the U.S. He's a very close colleague of mine, and so it's my pleasure to introduce him. Dr. Sharma is trained as a pediatrician and a reproductive immunologist, and his work has really been very focused on immunologic tolerance in pregnancy, and now is doing a lot of work on implantation and embryo development. So we're looking forward to hearing your talk.
Well, let me begin with thanking the organizers, Gautam, Allahabadia, Goral Gandhi, and others for the invitation. I have attended these meetings a number of times in the past, and I have enjoyed, and I'm sure you will all agree with me that these meetings are a perfect mixture of quality science, quality medicine, and fun part, that every evening that we have singers from all over India and presenting wonderful programs. So I think I consider these meetings to be a perfect platform for all of us to come together and learn from each other. So let me begin with my talk here. It's not going to be a talk where I'm going to tell you about ovarian stimulation or oocyte maturation or poor responders or higher responders or differences between recombinant hormones versus the natural hormones. This is a session, or rather effort, at coming up with some new, novel ideas, novel pathways to address the issue of embryo quality and health. Because you heard during last 2-3 days here that yes, we need to have proper ovarian stimulation, we need to have mature eggs, we need to have the right kind of hormonal stimulation, but the story does not end there. You need to go beyond from cell 1 embryo all the way to blastulation and implantation, and what happens beyond implantation. And that is the focus that we have currently, and what we are trying to understand. How do you provide? Because I have heard during this meeting for the last 2-3 days that yes, you have a mature egg, you can fertilize it, but that does not guarantee higher success rate. We still are dealing with very poor success rate in the IVF clinics. So our efforts are aimed at improving the embryo quality and health and the implantation. And what we are trying to do to come up with some novel ideas that might help us in that respect. So the title, From Implantation to Pregnancy and Beyond, Role of a Novel Protein, and I will be talking about that particular protein that we have identified that might be helpful in this process. Do we have a pointer somewhere? Thank you. So the main question that you have heard in this conference is this: IVF success depends on good quality and health of the embryo. And this is the question that we are trying to address. And this work has been done in collaboration with Dr. Jared Robbins and his research staff. When we started this work, we were totally confused what to do, how to address this question. And I was reminded of a quote by a famous person. This gentleman is Donald Rumsfeld. He was Secretary of Defense during Bush's presidency. And he was the main character for the Iraq War. And what he said once, and try to figure it out what he said. And this was the conundrum that we faced when we started this work. We know there are known knowns. There are things we know. We also know there are known unknowns. That is to say, we know there are some things that we do not know, but there are also unknown unknowns. The ones we do not know, we do not know. So try to figure it out, and the situation was as confusing as this statement this gentleman made. So let me begin with something that we learn in the medical school. And this is the basic fertilization process. And what this says that from ovary, we have heard a lot about ovarian stimulation, egg maturation. That leads to ovulation. Egg is fertilized in the fallopian tube by the sperm, and then of course it goes through cell division and blastulation, and then it implants in the endometrium. And if everything goes right, then it leads to a productive pregnancy. So this is the thing that we read in medical books, And this is what transpired for in vitro fertilization by Nobel laureate Robert Edwards. And basically, he tried to mimic what happens in vivo, retrieved eggs from ovary, and in vitro, they were fertilized, and then went through the process of cell division, and eventually with the hope that it will implant and everything will be okay. So this is the process that has also been adapted for in vitro fertilization conditions. What we are trying to do, is there a protein? And this is the question that I begin with. There are the current methods satisfactory to improve embryo health, implantation, and pregnancy rate? And my answer is no. We still need to really work at it, and we need to come up with some novel ideas to really improve the situation. And what we think that there has to be a protein or peptide or factor that starts out in ovary and could be participating in the egg maturation, then fertilization and cell division, and participates in the implantation and beyond implantation. So that is the goal that we have, that there must be a factor that regulates all these processes here. And this is where the story started. So let me preempt my talk, and this is the protein that we think is going to be very important for, and this is known as transthyretin, transporter for thyroxine, thyroid hormone thyroxine, and retinol. So this is the protein that I'm going to take you through with some experiment that we have identified that might be important. And what is thyroxine? Transthyretin? It is mainly synthesized in the liver and the choroid plexus in the brain, and it's secreted into CSF. For us, this was very important information. Estrogen stimulates transthyretin gene expression. These are some of the levels of this protein that are present in the CSF and in the serum circulation. And it's currently being used as a valid marker for protein energy nutritional station in kidney dialysis patients, and its half-life is 2 to 3 days. And this is the three-dimensional structure of this protein. It's a homotetramer, and this is the monomer molecular weight, 14 kilodalton, and 55 kilodalton is the molecular weight of homotetramer. Homotetramer is very, very unstable, so it has to be stabilized by thyroxine, T4. It can also be stabilized by non-steroidal anti-inflammatory drugs. Aspirin is a very potent stabilizer of this structure, and that may be one of the ways that aspirin might work for some of the things during pregnancy. So this is the structure. And what happens, this protein has been identified to be associated in a number of pathologies, and the main ones are right here listed: familial amyloid polyneuropathy, and what happens here, and Alzheimer's disease. Actually, it's in clinical trials for Alzheimer's disease patients. And what happens that this protein misfolds and undergoes aggregation, and it creates fiber-like structures that get deposited in different organs and cause toxic effects. And this is what is happening in polyneuropathy and Alzheimer's disease, because this protein, when it undergoes the misfolding and aggregation, fails to trap beta amyloid protein, which then forms plaques in the brain, and that is the pathogenetic pathway for the onset of Alzheimer's disease. So these are some of the pathologic effects of this protein when it undergoes misfolding and aggregation. And this is where we really identified this protein. Preeclampsia is a hypertension condition during pregnancy, and actually it manifests a number of pregnant women that have gone through the IVF clinics. Preeclampsia is one of the major manifestations in the IVF pregnancies, and that's where we really identified this protein. And preeclampsia affects 5 to 8% of pregnancies. It is associated with a high number of maternal deaths per year, and this is worldwide number, and it's a leading cause of prematurity. And there's no treatment for this syndrome. Delivery is the only treatment. Once baby is delivered, placenta is gone, and then person gets back to normal. So this is the preeclampsia, and if you want to learn a little more about it, I'm sure you all know about it already, but I just want to make two important points here. These are some of the diagnostic features for mild preeclampsia: elevated blood pressure, proteinuria, kidney pathology, glomerular endotheliosis, and not in all cases, but in many cases, these women also have edema. And then when severe preeclampsia, it can also result in HELLP syndrome here. And it also have manifestation on the fetal side, morbidity, mortality. It's associated quite frequently with intrauterine growth restriction, and it could also be associated with chronic diseases when these children become adults here. But the important point that I wanted to point out here is this thing. This syndrome has remained enigmatic, reason being we do not know what causes it, how to treat it. The only treatment, as I mentioned, is the delivery. So it still remains a huge problem, and it's a very serious pregnancy complication. So we wanted to learn about it, and this is where we really identified this protein. And this is one of the articles we have recently published on humanized mouse model of preeclampsia, where we have taken sera from pregnant women who are suffering from preeclampsia, injected that in pregnant mice, and we could recapitulate all the features that are associated with the human condition: elevated blood pressure, proteinuria, kidney pathology, production of anti-angiogenic factors, and hypoxic injury in the placenta. So all these features could be reproduced in a mouse model in response to the serum that we collected from women who were experiencing preeclampsia-like syndrome. And so this work is published here. The issue was what is present in serum, what is causing preeclampsia-like features, and that's where we have identified transthyretin by different proteomic approaches. I'm not going to bore you with all the experimental details. This work has been completed. It's under review in PNAS, Proceedings of National Academy of Sciences, USA. And the main features of this work, transthyretin is reduced in preeclampsia. Just like Alzheimer's disease and amyloid neuropathology, the transthyretin undergoes misfolding and aggregation in preeclampsia also. So it is a disease of misfolding and aggregation. And placental functions are compromised because aggregated transthyretin deposits into placenta and causes toxic effects. So this is the thing that, this is where we identified transthyretin.